In this animated sequence, we're going to look more closely at the hepatitis B viral particle and how it replicates. The hepatitis B virus consists of a viral membrane covered with surface proteins. The membrane surrounds a core particle that contains the viral DNA genome and the multifunctional HBV polymerase. HBV is transmitted via body fluids, either vertically at childbirth or horizontally from one person to another. The virus is present in very high numbers in the blood of an infectious individual, sometimes with more than 1 billion virions per milliliter. The virus can also remain infectious outside of the human body for at least 7 days. These characteristics make hepatitis B 100 times more transmissible than HIV. Once inside the bloodstream of the newly infected individual, the virus travels in the blood to spread from the infection site throughout the body, eventually converging at its target organ, the liver. Here, the virus enters the narrow liver sinusoids and passes through the holes or fenestrations in the sinusoidal epithelium to enter the space of Dis. From here, the virus can access its ultimate target, the individual hepatocytes of the liver. Let's follow this virus particle as it passes between the microvilli and infects one of these hepatocytes. The exact way in which the virus enters the cell is unclear. What is known is that the central core containing the viral DNA and viral polymerase ends up inside the cytoplasm of the hepatocyte. The core moves towards the nucleus of the cell. As it travels through the cytoplasm, the core begins to disintegrate, releasing the viral DNA and polymerase, which then pass through a nuclear pore and into the nucleus. At this point, the viral DNA exists as a partially double-stranded circular form. The host cell's enzymes then aid in several steps, including completion of the positive strand of DNA and covalent closing of the circle to form highly stable, covalently closed circular DNA known as cccDNA. During the course of the viral life cycle, more copies of cccDNA build up in the nucleus, forming a stable reservoir of viral genetic material within the cell. This cccDNA functions as a mini chromosome within the nucleus, serving as a template for cellular enzymes to produce new viral pregenomic and subgenomic messenger RNA. The viral RNA molecules pass through the nuclear pores to enter the cell cytoplasm. Here, some of the messenger RNA is translated by the cellular ribosomes to produce HBV core proteins, shown here in green, and HBV polymerase, shown here in yellow. The viral polymerase has 3 functional activities: priming, reverse transcription, and DNA synthesis. The principal function of the polymerase is to convert pregenomic viral RNA into viral DNA for incorporation into the next generation of hepatitis B virions. To accomplish this, 3 sequential steps must be completed. Firstly, a priming step initiates the process of negative strand DNA synthesis. Then, the negative strand of DNA is generated by reverse transcription with degradation of pregenomic RNA. And thirdly, the complementary or positive strand of DNA is synthesized to result in an infectious DNA genome. This priming sequence is the key to the replication of viral DNA. Without it, no further DNA synthesis would be possible. The 3-nucleotide-long primer base pairs with the other end of the pregenomic RNA and serves as the initiation point for the synthesis of negative strand viral DNA by reverse transcription. As DNA synthesis proceeds, positive strand DNA synthesis. As positive strand DNA synthesis proceeds, it jumps back across to the end of the negative strand of DNA, thus circularizing the partially double-stranded DNA structure. While the new HBV DNA is being synthesized, core proteins assemble around the viral polymerase DNA complex to form new viral cores. Meanwhile, viral surface proteins begin to build up in the membrane of the endoplasmic reticulum. The newly formed viral core buds into the lumen of the endoplasmic reticulum to form a new enveloped virus. The virus passes through the Golgi body and into a secretory vesicle, which then releases it into the extracellular medium by fusing with the external cell membrane. From here, the new viral particles can go on to infect other hepatocytes and spread the infection throughout the liver.